Good morning, everyone. My dear colleagues, my friends, and my dear students, and all the participants unvisible. Hope next time we will do it visible. So I have the very great pleasure of introducing Dr. Ramchand, C.N. Ramchand, a great friend of mine. And I think I can talk about him for half an hour, so I don't want to steal his time because he is also a person who will talk a lot of things. So I would like to say that Dr. Ramchand is from MS Baroda. He did his PhD with the father of our Nobel laureate Venki Ramakrishnan. So he did his PhD with Ramakrishnan. And after that, he had a stay in England. Of course, I think he is more English in certain professional things. And he has served as a vice president of research in Sun Pharma, Spark. And then he also did more or less the managing director, vice president of few pharma companies. And but he has in his blood that Gujarati training. So he became more a businessman than anything, but a scientist, a business scientist. So that's great, very rare to see such combination. So he has been creating structures with innovation because he is a person, I think his appetite does not stop with anything. He was voracious in creating structures and successful, not just creating and leaving like that, like some people, but very successfully. So he has started a company called Succeed just a few years ago, maybe 6 or 7 years. I remember the start, and it is going very, very successful. And I'm very proud to say that one of the very rare companies who has come up with a CGMP fabrication for the clinical trial within 2 months. So I had the pleasure of being in those, examining those things and finding it extraordinarily good. And not only Succeed, he has created another structure in Chennai itself, where it is a few companies and training. Training because he has understood the need that the manpower in India is not adequately trained in the right way to serve the industry in the what you call the good science with production. So, and this is what I think one of the very rare opportunities that he has created these structures, and he is also part of many companies like MagGenome, and MagGenome was the one who got the award. So I think I can go on talking, so I don't want to take away his time of talk. So, and also one more thing, he has an infectious disease, which is enthusiasm and energy. So he infects me all the time with that. So I want that he just give it to everybody, and instead of Corona, we'll take this as the new thing. So we'll take it from him, that enthusiasm and dynamism and energy. So on to you, Ramchand.
And I'll just share. Good morning, everybody. Good morning, those who are in India, overseas. And my special thanks to Professor Vijayalakshmi and Professor Krishnan. She was telling about, I have an infectious disease. I felt she is more, because at this age, I don't want to tell her age, but then she's the most energetic person ever I have seen. And, you know, she does not tell no to anything. And that's why, because of her, that great place CBST has come up. And her footstep, Professor Krishnan, also has initiated many things, especially back-to-back 2 conferences, Zoom conferences attended by 100 to 1,000 people all over. It's not an easy thing. So thank you both for giving an opportunity, especially the earlier also I gave a talk on therapeutics as well as diagnostics. Now it's all about drug discovery development of novel chemical entities, drug repurposing, immune modulators, and reverse pharmacology, which I will be giving. I will briefly talk about the companies what we are involved. Mostly it is in Chennai. We have facilities also in Cochin. Now, why I'm going to talk on corona, because we have some connection with the corona, though coronavirus and its understanding about the proteins have very recent thing, but then certain things we have to do. If you see, the Saxon Life Sciences is a very successful biologics company. We have developed a molecule for macular degeneration, which is in the phase 1 trial, just going into a phase 1 trial. Theragen Biologics is another biologics company. We have developed a molecule for diabetic retinopathy. So both these companies are based on synthetic biology. And synthetic biology is one of the most recent methodology developed for the drug discovery development or biologics discovery and development. And it's very essential for therapeutic development of the vaccines or antibodies or Fab molecule for whether it is coronavirus or similar molecule. MagGenome is other successful company. We commercialized the products a couple of years back, now reached in such a way that we have break even. And also we got a big grant from BIRAC. So we developed few products for corona. One is that viral transfer medium, unique medium. The other thing is that we also got a funding for developing RNA extraction, which we are already commercialized. But then for the purpose of RNA extraction, we are waiting for the approval from ICMR. We are also developing a product from saliva RNA. So other companies are that, Theragen Life Sciences, which is an ANDA company, abbreviated new drug application, where we are planning to do certain repurposing. And the fifth one is SIPWISE, basically functional drinks we are developing. Maybe one or two slides I might be able to show you. And a curcumin-based drink we are introducing, and within few days' time, couple of months' time, an immune booster or immune potentiation drink. I will be briefly talk about that. And another one we are developing is an immunoglobulin Y. So for a throat spray, nasal spray, that is another thing. So we have some connection with the coronavirus currently. And that's why I thought, maybe I might be able to tell about how the drugs or NCE'S biologics are developed. Apart from this, we also have few other companies where I was directly or indirectly involved. We also have a biotechnology, biopharma skill enhancement program, which is funded by Kerala State Industrial Development Corporation. About 10 to 20 students every year we take, mainly training purpose in the various aspects of drug discovery, development, biologics discovery, development, etc. Yeah, sorry, it's moving now. This will be the overview of the talk that I am going to give. I'll briefly talk about drug discovery. And I think yesterday, quite a lot of people have mentioned about what drug discovery, but then I'm going to talk normally not on drug discovery per se, but I'll be briefly talking about how drug discovery can be done for COVID viral proteins. There is a difference is there. The reason is that it's a recent virus, and nobody knows about the protein. So you can't use the classical way of drug discovery over here. Everything has to be very quick. What can be done? The same way biologics, whether it is vaccine or antibody or Fab molecule, how that can be developed. Again, it has to be quick, within few months to 1 year or 2 years. Otherwise, there is no fun, because the pandemic is increasing and many people that, reaching herd immunity. So unless the drug is developed very quickly, it does not mean anything. Then I'll be talking briefly about the protein targets for control of COVID-19 and what are the targets in host cell as well as the virus protein. Then small molecules under development for COVID-19. Currently, you might have heard about many molecules are coming out. How this so quickly is coming out? What is the technology they are using? So I'll be briefly talking about that. The same way, novel biologics for targeting COVID-19. Then what exactly drug repurposing? What is reverse pharmacology? What is vaccine development and how it is done? Briefly for the coronavirus, and what is immune potentiation? Please remember, the slides are very complex, so I will try my level best because I know a lot of students are attending. So as simple as possible, I will try to explain. Now, NCEs and biologics discovery and development, what are the major phases? You all know that, the first is the research phase. Research phase is the phase or the discovery phase you can call. That is the time how the target protein is identified. What is the therapeutic area of interest? In the therapeutic area, how the therapeutic protein or the therapeutic target is identified. One of the most complex things ever mankind known is this. Because there are thousands and thousands of diseases are there. Still, we do not know many of the diseases. None of the neurological diseases or something connected with the CNS, central nervous system, brain, nobody knows. For example, schizophrenia, depression, mania, manic depression, Alzheimer's disease. 20% of the people are affected by Alzheimer's disease. Is there any drug? No, nothing, because the mechanism is known. So the research phase is the most important, especially novel target. The second thing comes once the target is identified, you will have to identify what is the drug, comparatively simpler than identifying a target, whether it is biological molecule or a small molecule. Now, the challenge comes the development of the molecule. Probably it is, for example, if you want to discover a small molecule or a biologics, is not that great difficulty, because once you have an idea about the structure, then you will be able to synthesize the molecule. Thousands of efficacious molecule you will be able to synthesize. But developing this molecule to reach the commercial phase is very complex. It takes 15 years. That is the challenge. So development is not that easy. Please do understand, only few hundred molecules are there for all the diseases combined together. So the major challenge is that. Then the commercialization phase, that is again, that's the job of the pharmaceutical company. Here, one thing I want to know is tell is that the major research phase is happening world over universities, and companies normally will not spend too much money on identifying novel targets because extremely risky, a lot of money is required. Now, if you see here, this is the drug discovery is complex and time consuming. Now, I gave this over here just to let you know, normally it's about 12 to 15 years and $2 billion is required for developing a drug, whether it's a small molecule for COVID viral targets or as a vaccine or an antibody. So if 15 years takes a drug to develop after conceptualizing, understanding the target, what is the fun in developing a drug for COVID? So you can't go through the classical way of drug discovery process. If you really see, there are multiple phases are there. There's a strategic research phase to understand about the target, what type of protein you should select, and how to identify the target and to validate the target. Then there is an exploratory phase in order to generate the hit molecule, then hit molecule to lead identification. Multiple things are done, whether it is virtual screening or assay development or high throughput screening. Then drug to redesign to get the best efficacy and safety, and finally the hit to lead molecule. Then lead optimization, and animal models is very, very important because most diseases do not have an animal model. Unless you have an animal model, you will not be able to tell eventually this is going to work or not. And that in the animal models also you can do not only efficacy, safety, but the absorption and its metabolism, excretion, distribution, etc. Then once the molecule enter into the clinical trial, it takes another 6 to 7 years. But let me tell you, all small molecule, 80% fail during this time. And during 10,000 or 5 to 10,000 molecules, 1 or 2 molecules enter into the clinical phase. I just wanted to tell you the complexity in drug discovery and development, then why we should go and do a drug discovery process for a pandemic like coronavirus. That's what I want to tell. So the classical way cannot be done. Now, I'll also mention about, drugs and biologics discovery and development. What are the different types of chemical or biologics entities available? Let's say that, let's go through that. If you talk about biologics, there is novel biologics. If the target is absolutely novel and your antibody or the Fab molecule or the CDR is novel, that is called novel. And another one is novel biologics, or it can be also called biobetter, in which the target and mechanism is same, but CDR, complementarity determining region. This is the paratope which goes and binds to the epitope, or rather the antibody goes and binds to the antigen. That will be different. And there is something called biosimilars. India, it is very popular because whatever is the novel drugs, novel biologics developed in US, Europe, we develop it as a biosimilar. But biosimilar development also is not that simple. Exactly similar pattern of the sequences, its metabolism, configuration, conformation of the protein has to be there. Now comes small molecules. Please remember that not many NCEs are developed in the world, only 420, and there is another category called Me Too drugs based on the existing NCE, a structural analogs. These are called Me Too drugs. So India has not developed the NCE, only 7 countries in the world could. So NCE is another one. ANDA, abbreviated new drug application, which is mainly that drugs or generic drugs that is to be introduced European US market, that has to go through a very different process, comparatively difficult, takes about 3 to 4 years or 2 to 3 years. Then comes generic molecule. Most of the pharma, Indian 26,000 pharmaceutical companies are there. Out of that, 99% of the companies are doing generic molecule. What is new chemical entities? You can just see a protein and to which a molecule goes and binds. I have shown some simple structure in the left-hand side and how this goes and binds. A molecule with a novel structure that is newly identified as potential drug, and that is going to be the key ingredient. When you are developing a drug, the structure, there is some requirements are there for the structural aspects of the molecule. You can't develop the pharmacophore for anything and everything because the molecule should have very good bioavailability. It should go around the body, enter into the cell, only go and bind to the target protein. So it's a very complex process, and the interaction normally with the target protein and your drug is multiple. It is hydrophobic or hydrophilic, electrostatic, hydrogen bonding, van der Waals forces. So multiple forces are involved in, and this binding has to be reversible. Now, designing NCEs and biologics is very complex also. It's not very simple because the specificity is very important. You can't go the drug or the biologics to go and bind here, there, then it will develop toxicity. We have to be very careful that the molecule should be absorbed or administered properly. It can be an injectable or it can be given orally. Then, it should be distributed throughout the body. There is a binding process to the body proteins are there. That is the way it is distributed. It should be appropriately metabolized and excreted. Otherwise, it will become toxic. It's very important that, to reduce adverse effect and improve efficacy. And it's also to understand the right dose, right drug, and the right time. What time to be given, once in a day or 4 times a day. These type of things has to be there. So these are, and it is also important, FDA or no regulatory people will approve without understanding the mechanism. Because what is the drug structure, where it goes and binds, what is the transduction process, everything should be. It takes several years. And it is challenging too, especially the development process is extremely challenging. What is challenging means, the uncertainty of development. If you are developing a biologics, Professor Vijay knows that, the complexity, bioseparation, the molecule should be separated with all the other toxic substances or other proteins. Scaling up, Because from lab, like 100 ml or 200 ml to 10 liter to 100 liter, finally to 2000 or 5000 or 10,000 liter, lot of problems you can face. Analytical challenges. For example, if you are developing a protein, there is at least 20 different analytical tests is there. One is its configuration and multiple conformation, the formulation challenge, then the stability of the molecule, then the multiple assays, surface plasmon resonance if you are talking about an antibody, or the antibody's affinity. So all these has to be that these are the challenges during the development process, and all has to be sorted out. That's why it is extremely difficult to develop, whether it is biologics or an NCE. That's why very, very few companies could develop that one. Let me also tell you that we are the only single company who has gone through all the RCGM requirement to get into a phase 1. One of our molecule is reached the phase 1 trial in totally novel biologics, which is against VEGF, vascular endothelial growth factor, for macular degeneration. And it's a very time-consuming complex. Now let us see about the viral, how we can develop drugs against virus. Please remember, if you are talking about the human body, all the targets we know very well, and it has been last 100 years or 70, 80 years, this has been going on. Now, many protein structures, configuration, conformation, three-dimensional conformation, its transduction process, everything is established. So what we need to do is that develop the drugs against that. But then that is not the case with the virus. Suddenly a virus has come and the pandemic has started. You can't develop a drug against that. So what is to be done? Now the scientist has a methodology immediately to sequence and predict the structures, and you can go through X-ray crystallographic structures, and that is what is done. So there are methodology to understand the structures, these novel structures of the protein. That has been done. Now, if you see about this virus, what the virus can't replicate by itself. It needs other organisms like a bacteria or a host cell is required. Now, these protein, there are not too many proteins. If you are talking about the human body, which we have about, we have about 25,000-30,000 genes and probably so many numbers of protein. But in the case of virus, probably 60 or 70 RNA is there. Probably it produces 30 or 40 proteins, and many of the proteins are inside. Why I'm telling these proteins are inside, it's not easy for a small molecule to penetrate, go inside and work against the protein. So it's not possible. So you will have to find out what are the proteins which is available outside so that the molecule can go. And few proteases are there. So mainly these virus contains some of the functional protein. There is structural proteins are there surrounding that, but it is not easy to target a structural protein. The reason is that structural proteins do not have a binding site to alter its function. But if you see about proteases, it has got very clear-cut functions are there. There is papain-like proteases are there. Then there is other few proteases are there. Then there is RNA-dependent RNA polymerase. These are some of the 3 or 4 very good targets which has been identified that can be used as a drug target. Now I have also given that antiviral activity targeting the viral metabolic function, which these 2 proteins, proteases and RNA-dependent RNA polymerase is involved in the metabolic function. The other thing is that you can also target the host cellular function. Why host cellular function? Because as I mentioned, the virus has to interact with the host cell. One is that, it interacts by going and, interacting with some of the surface protein. In the case of coronavirus, there is a enzyme called angiotensin converting enzyme, which acts as a receptor. And there is a spike protein surrounding the virus. That protein goes and binds to this angiotensin converting enzyme. And after binding, it goes inside. A fusion takes place, goes inside. Once it goes inside only, the viral replication can take place. So if you inhibit that process, it's possible. and the target host cellular function of that particular protein, angiotensin converting enzyme. Already few drugs are also there. So how this can be used? The same way RNA dependent RNA polymerase that is involved in the development of certain other proteins within the host cell, and how that can be inhibited and the virus can be killed. So these are the things what the scientists are working on. So this is basically some of the protein targets that I have mentioned. If you see what you are seeing above, this is the coronavirus, and these are what is spike protein. And you can see one is the spike protein interacts with the angiotensin converting enzyme, and once it do not interact, it cannot go inside. So this interacts, and one is this particular interaction allows to fuse with the cell and goes inside. Okay. Now I have also given a drug that Ramipril, which is basically, I think it is a hepatitis C. Sorry, it is a blood pressure. It is involved in the inhibiting angiotensinogen to angiotensin. So it's a blood pressure drug. Now, this has been shown to bind to angiotensin converting enzyme. If this converts, if it inhibits, then this interaction will not be there. So I think Ramipril is already a repurposed drug. Now, if you see, I've also given some other protein. This is a host cell. It's a protease. This protease is involved with the activating this angiotensin converting enzyme. So this means that, unless this protein is there, you will not be able to activate this angiotensin converting enzyme. So there is already a drug called the chemostat, which is also currently being developed or under development to inhibit this particular protein. And once this particular protein is inhibited, the virus will not interact with the angiotensin converting enzyme. So this is another repurposed drug. The third is CD147. CD147 is another drug, another surface protein, which is otherwise mainly involved in the purpose of, CD147 is mainly involved in the purpose of certain diabetic complications, and another one is connected to, I think, asthma. So there are few drugs are available, mainly antibodies. I think it is under development. So as I mentioned, the diabetes and asthma, and there is another molecule called furin. So there are few molecules is under development. So I just wanted to tell you one thing, that both the virus as well as the host protein, there are certain targets which is already identified. The molecules are there. This means that you don't have to go through the very complex process of discovery and development. That means probably 3 months to 6 months time, the drug can be introduced, because many of these drugs are already tested in human. This is what is called the repurposing. It's very, very important such methodology to be used, because you can't wait for such a long time. And what you are seeing is that, multiple protein, whether it is spike protein or membrane protein, envelope protein or nucleoprotein, probably can be used as a target for the development of vaccine. Mainly spike protein is used as vaccine, but many of these proteins can't be used as a small molecular drug, because these are all structural proteins. Now, ideal protein targets in SARS-CoV-2 VIRION, one of the molecule is spike protein. Okay. Now, spike protein, as I mentioned, basically it is involved in interacting with the angiotensin converting enzyme. So if you see here, there is a good interaction. What you are seeing here is the complete, above this, the complete RNA sequence details, which is about 70 different RNAs are there. And down you can see multiple proteins, and some of these proteins are certain functional proteins like papain-like protease. Then another protease, which is 3CL protease. Then RNA-dependent RNA polymerase, and certain ribonuclease. So these are some of the proteins which has already been used to repurpose existing drugs for that. But S protein, I suggest, I think mainly can be used for the purpose of developing antibodies or vaccines too. Now here, this is an interesting slide which shows various drugs developed or under development that can be used for targeting coronavirus. As I mentioned, there are 3 phases are there for the virus. One is the virus has to fuse. Then in order to fuse with the cellular membrane, go inside, it needs another target within the host protein, which is angiotensin converting enzyme. Once it binds to angiotensin converting, it fuses, goes inside. So as I mentioned, there are few drugs that can inhibit this process. Second one is endocytotic process. Now, endocytosis can be inhibited by chloropromazine. Very, very interesting. I was thinking about this particular opportunity with chloropromazine. I just went through the literature. People are already working on that. Chloropromazine, as you all know, is one of the most important antipsychotic drugs, but it was never developed as an antipsychotic. It was originally about 70 years back developed for the nausea and vomiting in pregnant women, the very first one, the GABA receptor. Subsequently, it was repurposed for anti-H1, for that is mainly used for allergic purpose. Subsequently, this has become an antipsychotic drug, which is dopamine D2 receptor. Subsequently, in 15 years back, it has been used for diarrhea purpose. That means it has been shown to inhibit the cyclic AMP, the production of adenylate cyclase. So inhibits adenylate cyclase. Now, this has already known chloropromazine has a membrane stabilizing ability because of its hydrophobic, hydrophilic, as well as charges. So now this is has reached the phase 3 trial, chloropromazine, for inhibiting the endocytotic process of coronavirus. So you can see over here multiple ways by which a certain existing drugs can inhibit. So one is the transport of the virus inside the cell. After reaching that one, there are 3 host cell proteins are there, already existing drugs. Everybody heard about chloroquine, how that inhibits, but I'm not sure that how effective that one. But scientifically, that structurally it is possible. But then it is very, very important. Unless this is proven in a clinical setting, it does not mean anything. This is the unfortunate thing, that we can, scientifically, in vitro, lot of things can be brought about, but it has to be proved in a clinical setting. But let me tell you, I don't think there are at least 5 or 6 drugs are there which is not very, very clearly proven that this can be used for coronavirus, though people are using that one. So maybe after a few years or a couple of months, we will know very clearly whether really it is effective or not. So chloroquine, then chloropromazine, then tetraandine, another drug. Then there are few other drugs are also in the process of development over here. Now, the second is once it goes inside, what is happening is that the virus will have to get involved with the host cell, the DNA. Then the replication takes place, which is the main enzyme is RNA-dependent RNA polymerase, and there will be a genomic RNA will be produced for the replication purpose, and which produces subgenomic RNA, which is translated into proteins, combined together, the virus will come out. This virus will be later used for the production of the adaptive immunity within the host cell or within the animal. So this is basically the thing. What I'm just showing is that how the virus is transported inside. After entering that, what all things can be done. Let me tell you, all these drugs currently being tested or already introduced, but Remember, everything took only 3 to 4 months time compared to 15, 16 years time. This is the power of repurposing the existing knowledge we are using, because at this point, nobody can think about developing a drug and, and test it. By that time, the pandemic will come, herd immunity will come, everything will become useless. So this is another molecule currently being developed. It is a nucleoside, ribonucleoside analog. So you can't introduce a ribonucleoside itself, because always, if you want a stable molecule within the body, it has to be analog. So this is an analog of ribonucleoside, and it attacks RNA-dependent RNA polymerase under development, but which is a new molecule. So people will know very soon that how this is going to be effective. Now, if you see over here, I have mentioned about 3 different proteases, which is coronavirus proteases, that is being tested for various drugs. And probably this might be using, I'm not sure whether indinavir, which is an HIV protease inhibitor, then ketoamide, and another one is GRL0617, probably under development. So within few months time, it might be used. But if it's a new synthetic drug, it's not going to come very soon, because you have to go through all the phases of drug discovery. If it's an existing drug, it is repurposed. Maximum time required is 2 to 3 months time. Now, what is novel biologics? Now, that's what we are doing. Novel biologics is, it can be an antibody, a full antibody, part of the antibody, Fab molecule, single chain molecule, or the molecules can be combined together. The Fab molecule can be combined together to interact with multiple targets. Now, classically, I have shown here that, that the antigens are injected into animal, and there is a process to develop the antibody. It was not very successful because you can't produce very effective molecules this way. Though earlier some vaccines can be done successful, but no antibodies can be done. But things have changed. It's by synthetic way. One is using a technology called phage display technology can be used. That's what we are using. Subsequently, all this DNA can be synthesized. DNA can be synthesized, can be introduced in the plasmid. Then, transcription, translation, and your protein can be generated within the host cell, bacteria, mammalian cells, and everything. So by using this synthetic biology, you can now synthesize anything, including a cell. You will be able to develop basically a cell. For example, membrane, if you want lipid can be synthesized the same way, every DNA, every RNA, every protein. So that's why it is called synthetic biology. Synthetic biology is coming up in a very big way, but currently extensively use the synthesis of DNA for the recombinant DNA technology, and nobody is going to use the classical way of producing antibodies. But there is a very clear-cut difference is there between when you are developing a small molecule or a large molecule. Small molecule is, certain aspects is very complex in small molecule development, because thousands and thousands of molecules has to be synthesized. Because it is coming from outside, synthetic molecule, you can never predict about its toxicity and pharmacokinetic profile, and it can give toxicity because it can go and bind to many other molecules. So on the other hand, biologic molecule is not like that. Complex in developing, especially that in order to get appropriate yield successfully for commercially successful product to develop. So major differences are, low molecular weight, large molecular weight, or usually organic or chemical synthesis made with recombinant DNA technology. Few critical process steps, but many critical process steps and in biologics, well characterized, less well easily characterized because of the conformation of the protein. Configuration is well characterized, but then what are the types of bonding? What type of hydrogen bonding interactions? It's very difficult to predict and develop. Non-structured structures may or may not be completely defined or not. homologous drug substance, homogeneous drug substance, heterogeneous mixtures. Many of the many of the molecules can be heterogeneous, especially when you are talking about, certain vaccines and other things. Normally, small molecules are not immunogenic, but this can be immunogenic, especially highly humanized molecule. When you are giving to animals, this can be immunogenic. So otherwise, if it is not fully humanized, then when you are giving to humans, it will be a problem. It can be immunogenic. I briefly mentioned about synthetic biology. So if you see the left-hand side, you can break down an antibody and make it to the multiple Fab structures or, constant region. But that is no more done now. It is all done by synthetic biology. And you can link, for example, 2 Fab molecules like this. And one CDRs can target to one target. This can go into another one. Now, if it is a trifunctional antibody, if you want to, for some reason, that is possible. A single chain can be made. So any different types of antibody fractions, small molecule. So there is significant increase and improvements has happened in synthetic biology. You might be able to see a lot of interesting things in the future. So this is a molecule that we have, involved in developing. If you see, this is an Avastin, full 180 kDa molecular weight. You can see here light chain, heavy chain. This is a constant region. What we have done is that this part we have synthetically developed. So if you see over here, so this is basically interaction of a Fab protein of Avastin to VEGF, but this is our molecule. Sorry. Yeah. So what you are seeing is that, this is a dimer of vascular endothelial growth factor, which is a dimer. And these molecules are Fab molecule, and how it is connected to VEGF and how it is neutralizing VEGF. That is what it shows. VEGF dimer binds to, for example, ranibizumab. Ranibizumab is Lucentis. I'll just briefly discuss about what exactly is drug repurposing. I briefly talked to you what is repurposing earlier. A typical example is chloropromazine. Now, if you see the chloropromazine, all the evidence has come from the clinical or during the clinical testing, clinical evaluation. As I mentioned, chloropromazine from the beginning, 70 years back, now it is used for corona purpose. So 5 different applications are there. Now, for example, 15, 20 years back, if you have mentioned that, this drug goes and binds to 15 or 10 different receptors, probably it would not have been, FDA would not have been granted the approval, probably assuming that this might develop some toxicity. There can be problem. But anyway, it does not have toxicity, and it is a repurposed drug. So normally repurposed drugs include approved, investigational, discontinued, or shelved therapeutics. So you can use an existing drug, which is off-patent drugs. Sometimes it is investigational. There is a commercial requirements are there to do that one. Many times discontinued drugs. What will happen is that the drugs during the efficacy trial might fail. It would have never reached the trial if it was showing toxicity or problems with the pharmacokinetics. So a drug without a problem with the pharmacokinetics, low toxicity, can be used for the repurposing. There are a lot of shelved therapeutics are there. So an universal strategy for neglected disease. What is neglected disease? There are, as I mentioned, 35,000 human diseases, about 25-30% is neglected disease. Neglected disease in the US FDA terms, people suffering from 200,000 or less. For example, this is a pandemic, it can't be a neglected disease. Schizophrenia is not a neglected disease. Diabetes is not a neglected disease. But diseases like Tourette syndrome, very rarely happening, a neurological syndrome. So that's a neglected disease. So in neglected disease, repurposing is a very good strategy. Or diseases like coronavirus infection pandemic, which you have to develop a drug very quickly. So what are the ways by which is done? Pre-existing drugs, where there is a clinical evidence is there, very good clinical evidence, FDA approved with the similar structures, or FDA approved drugs with side effects can be used. Then failed drugs, but failed in terms of efficacy, not by toxicity. Of course, we can't go ahead with a drug with toxicity at all. Now, what are the concerns when considering a drug for repurposing? Is it off patent? Because if it is still under patent, you need to negotiate with the company which owns the patent. Is it FDA approved? If it is an FDA approved, your whole process will become very easy. Is there prior art for it being used in new indication being studied? Most important thing is that if you want study and original patent, the medical application has been, already been covered, then you won't be able to do. You will have to negotiate with the company. Is it still being made? It's very, very important that you know whether the drug is available. Otherwise, you will have to start synthesizing the molecule. Does it work in a relevant animal model for the disease being studied? It's important that if you are going to understand about the new mechanism, then is it possible to, or the animal models are available. Does it work better in combination with another repurposed drug? That is also can be done. So two molecules can combine together. But people are using a lot of collaborative effort to develop molecule. Let me tell you, it's an extremely good thing, repurposing of drug, good science, good bioinformatics scientists is there, because all these complications of 15 years, but the value can be generated quickly, but you know, a very good scientist, a teamwork is required developing repurposed drug. This is a very typical example of repurposing of existing drug for coronavirus. Left side, you are seeing a target, which is a protease, I think. The right side, another RNA-dependent RNA polymerase. Left side, hepatitis C drug, remdesivir. And you know that that is very much used now. Now, what was originally remdesivir is used? Hepatitis C. I think it is last 15, 20 years it is being used for hepatitis C. But now suddenly, the scientists have found that, you know, this particular molecule can bind to a protein which is present in coronavirus, which has got homology with hepatitis C, that particular target. That is the way they came across. And after screening, they identified that one. The second thing is that an anti-HIV drug, lopinavir. So lopinavir is mainly RNA-dependent RNA polymerase. I think that is being tested or introduced in the market already. Now, drug repurposing does not take too much time. As I mentioned, if you have evidence, one is clinical evidence. The second possibility is that you can screen using docking studies, because most of the protein has got X-ray crystallographic structure. All details are there. In vitro or in silico, you will be able to screen and develop novel structures, but the drug is already tested, no toxicity, and clinically available. You don't even need to do a phase 1 trial. Now, normal time, they will ask for a phase 2 and a phase 3 trial. But at this time, if you are developing against coronavirus, probably immediate approval can be there, so that, you know, a small phase 3 can be done, and then they will introduce in the market, and so that, you know, if it is going to be useful, is a great thing. So normal repurposed drugs is about 2 years against about 10 to 17 years for the normal drug discovery program. But the current phase of this pandemic, many of these drugs are getting approval in 1 to 3 months time. Straight away they are giving approval, but they know the dosage is not tested at this time. The dosage they are using, the existing dosage. So that's one important thing, because it all will take time. If you want to establish the right dose, for example, remdesivir, I don't think they are doing a new the dosage for that. Probably currently using is that existing dose only. So I'm just in a cartoon format, just mentioning how drug repurposing works. It is normal drug discovery, very complex, goes through all the processes. On the other hand, in repurposing, especially clinical evidence and existing drugs, it is very quick, probably few months to 1 or 2 years time. Now here you can see currently some of the approved drugs. I mentioned what are the targets, whether it is mainly involved in binding to the host cell protein or the transcription translation after going inside and combining together, coming out. So any of these targets, whether it is viral target or host cell target, can be involved. So there is nymphomostat and chemostat. Mainly involved in, the first one is a protease inhibitors. The second one, chloroquine, is angiotensin converting enzyme. The other one is chloroquine and hydroxychloroquine, that is also involved in endosomal acidification process. Remdesivir and flavipiravir, which I already mentioned, which is RNA dependent RNA polymerase. Lopinavir and ritonavir and disulfan. So these are also proteolytic enzyme inhibitors. There are a few more drugs under development, cyclosporin, which is an otherwise immunosuppressant. So what I just wanted to tell is that there is few repurposed drugs currently available. The repurposing is comparatively easier, quicker. Within a couple of months' time, people will be able to develop. But you need to go through the clinical evidences, and also the structure of the protein of the virus or host cell is very, very important in order to prove and get into the clinical trial. So this is basically the mechanism of remdesivir, how this is happening. For example, after seeing this, I think that a combination of angiotensin converting enzyme inhibitor along with remdesivir could be a good opportunity. By the way, remdesivir is a prodrug. That means once it goes inside, part of it is broken down and become active drugs. You can see the right-hand side is the active part. This is the prodrug. There is a hydrolysis takes place here. This ester bond is hydrolyzed and you get this molecule. Once this goes inside, the translation of the viral polymerase RDRP, is taking place. Then this is exactly what is happening within the cell, host cell. So a genomic replication and the RNA is produced, which produces subgenomic or transcription process. Many of these proteins are formed. Now all these things combined together, the virus is formed within the host cell. This comes out. Then this is getting involved in its replication again, and also during this process, this will be attacked by macrophages, antibody will be done. Now, the question is that if your antibody is the antibody production is immediate, very quick, if you have an extremely high immunity, you don't have to worry. If that takes time or your innate immunity is not taking care, there will be a problem. A few more slides. I will very quickly finish. I know the time is getting very closer. What is reverse pharmacology? Another approach, something similar to repurposing. Reverse pharmacology is that in which the classical way is that, right from target identification to lead optimization to preclinical studies and clinical trial, which takes several years. Now, if you have an evidence, and normally reverse pharmacology works well with classical phytochemical or the other Ayurvedic type products, it can be used. But the first thing is that the clinical evidence is required and you should be able to enter into the clinical trial. Now, if a new molecule is there, it takes years and years to get into clinical trial. Already, suppose you have other evidences are there in the clinical evaluation, then you can test for other applications. Yesterday somebody was mentioning metformin is a classical example of such a thing, that in which metformin is originally used for diabetes and few other things. Now it is used for certain cancers like breast cancer. You all know about another very good application is this drug which is involved in inhibiting platelet aggregation, which is aspirin. Aspirin is another repurposed and using reverse pharmacology approach which has been done. So what I want to tell is that, you know, reverse pharmacology approach is an extremely good technology where if you have certain evidence and you can go into clinic, then you can study the pharmacological part, then where it binds, what is the mechanism, all can be established, and probably 1 to 3 years time the molecule can be done. This is a very good method for developing drugs or at least active ingredients from Ayurvedic or plant-based products. See, plant extracts, if you want to test in human, as long as it is not purified, you can do under very easily. If it is, as long as an extract, if it is being used, then you can study whether it is in animals or humans, which disease it can control. If it really controls certain disease, then you can study further that extract, purify the product and develop it as a drug. So just I wanted to tell that, you know, reverse pharmacology is an excellent method. So I'll just briefly tell about the vaccine development. All of you know about that one. What is innate immunity and adaptive immunity? Innate immunity is very important. I just wanted to tell what is immune potentiation. Really clear evidence is there or not. I wanted to tell that one. In innate immunity, you know about physical barriers. For example, your skin is a physical barrier. Your nostrils, there is some epithelial, layers are there. If you go to your stomach, there is acid is there. These are all physical barriers. Now, what we are trying in this time, the most successful method is the physical barriers. Currently, we are using, either we are using a mask. If it is not the mask, social distancing, or washing the hands continuously so that, you know, organism is there. Everyone knows that this is a very successful method. There are chemical barriers are also there. Chemical barriers, for example, in your saliva, there is a proteolytic enzymes are there. For example, if it goes into the mouth, that proteolytic enzyme or lysozyme. For example, in the tears, lysozymes are there. If an organism goes, virus goes, that can kill the virus. So these are all very good methods. But activating this or increase this activity, I don't think anyone knows. Then once this virus somewhere or other goes through our physical barriers, goes inside, we have many other methods by which within the cell, within the few hours, you can see. These are NK cells are there, dendritic cells are there, basophils, eosinophils, neutrophils. These leukocytes can immediately kill by multiple methodology. So now, innate immunity, if you have an extremely good innate immunity, that will protect you. Now, who has very good innate immunity and who do not have, how to activate that one? That's a billion-dollar question. Because people, you will be able to see hundreds and thousands of products. People are telling to take vitamin, people are telling to take minerals, people are telling some Ayurvedic product, people are telling exercise is good. Some people are telling that, you know, exercise with your stomach. Some other people are telling pranaya is good, or, you know, your the respiratory control. So many people are telling, and whether you are doing swimming or running or jogging or walking. So many things. Some people are telling very good nutrition, but let me tell you, nobody has proved that which actually, because if it is to be proved, it's not in vitro. In vitro, many things can be proved. In a clinical setting, under controlled condition, blinded condition, this is to be proved. For example, someone is telling you do this one, your natural killer cells can increase significantly 20%, 30%, then it is understandable. Until that time comes, or someone is telling that ginseng can increase so much. In vitro is fine. I worked on curcumin. Curcumin has got 11,000 applications, probably almost 40. I myself have got about 30 to 40 patents are there. One of the first highly bioavailable curcumin is introduced in the market, but not yet proven clinically. Unless that is proved clinically, it does not mean anything. Now, adaptive immunity. There are two ways.
Ramchand, I think you have to wind up. Yeah.
Okay. Yeah. So probably, I think then, can I have 2-3 minutes?
2 minutes, 2 minutes. Because it's very interesting, extremely interesting. But unfortunately, time is a constraint.
Okay.
Go ahead.
Yeah. In adaptive immunity, you have your helper T cells, cytotoxic cells. That will, few days it will work. But the major thing is that if you are attacked by the virus, then your immune cells will take over. Then antibody will be produced. There is a memory cell as well as, plasma cells. Plasma cell will secrete your antibody. Your antigen or the virus will be immediately neutralized. Then you get an attack later. Then your plasma cells will memory cells will release the antibody and you will be safe. So I'm not going to go ahead with any of my slides. Just I want to tell that many of the vaccines are under development, whether it is attenuated or inactivated or weakened virus can be used. Viral vectors, it is being successful in animal vaccines, not yet in humans. The same way nucleic acid is under development. Most successful is that protein-based, whether it is protein subunit or virus-like particles. So inactivated and weakened, very successful. Protein-based, very successful, currently under development, whether viral vector or non-replicant. So these are the different types of virus under development. So vaccines, I know how it is developed. If you have a weakened virus or inactivated virus, it can be introduced. Then what will happen is that your immune system will take over by dendritic cell or macrophages that will interact with T cell, B cell, produce your immune antibodies, and that can kill your virus. So these are some of the major vaccine in development. I just already mentioned about what is immune potentiation. Now, immune potentiation is that whether is there any real drug or a supplement really to potentiate, increase your immunity. There are hundreds of products are there in the market. Constantly it is coming up. You know that, in the airport now everybody is taking curcumin-based drinks. Is there a proof? If there is a proof, you need to have clear clinical evidence. Until that time, there is nothing wrong in taking. We will have to assume that it increases this thing. For a scientist to believe, you need to have real clinical evidence. So I'm not going anymore. This I've also given a slide on ginseng. So many people world over is using, but most important thing is that these are all in vitro evidences, but clinical evidence is required in a clinical setting. So, and we are developing another antibody-based therapy where it is called immunoglobulin Y. That's actually in collaboration with a US company. We are in the discussion process. You can produce immunoglobulin called IgY, or in the yolk it will be produced. Then this egg yolk can be used, your antibody source, and a very interesting technology. And we are trying to develop as throat spray, nose spray, a drink, etc. So I think, thank you very much. And yeah, thank you. Thank you very much.
Thank you. Thank you, Dr. Ramchand. I think extremely rich. I don't know. You are very good in making companies and financially strong and also scientifically too rich. So I think I have a very quick question before I give it to the participant. See, in the drug repurposing, when you compare small molecules versus protein molecules, how would you say in terms of time limit and the usefulness, which will be the, I think, obviously it must be small molecule, but I want your comments on that.
Yeah, see, normally repurpose is very successful in small molecule. The problem with large molecule, fab molecule,
I was
the
going to say
issue is that it is highly specific because CDRs are highly specific.
And also the three-element structure will just go and bind somewhere. That was the purpose I was asking the
Correct.
question.
With the same target, different application is fine. Something similar to that. For example, let's say in our case, VEGF is involved in multiple
Yeah, I know.
opportunity. So wherever that can be done, one is it is involved in any angiogenesis process. Anywhere angiogenesis is used, it can be used. So that is one way of doing, and it can be used, biologists can be used. But the real repurpose is that small molecule, multiple targets, identifying multiple targets without any toxicity.
Thank you. So I think maybe some questions.
I will request Dr. Sabris to take a few questions from participant.
Yeah, thank you, Sanjit. A few participants have asked questions. One participant is asking, in drug repurposing, how do we decide the dose of the old drug for new activity?
Excellent.
That is what he was telling.
Yes, which is, I mentioned that one. See, currently under this condition, because, you know, if you need to identify the right dose, it will take time because we need to evaluate. A typical example, as I mentioned, is aspirin. Aspirin is a COX inhibitor, which is also a leukotriene inhibitor for platelet aggregation. In the first case, it is 200 to 400 milligram. In the second case, it is 50 milligram. It takes some time to establish the dose. So, but there is nothing wrong that, you know, at 400 milligram also, it can be a platelet aggregation inhibitor, but it is too much. So, but, you know, in an emergency, you can use. So what is companies are doing in an emergency, existing dose is used, but during the process, they will be able to come out with the right dose.
But there is one problem. If the existing dose is not efficient, then you have to increase. So that where will be, we will have a hurdle.
Right. If the dose is, for example, I had exactly a similar thing for hepatitis C when we were developing. The dose was about 3 times. The Indian regulatory did not give us approval to go for hepatitis C.
That's a very important problem. Yeah.
They told we will have to do a dose response phase 1 trial. It is as good as any other trial. But then we did that in Belgium. This is for chemists.
Okay. Thank you, Dr. Ramchand. Another question from the participant is, can adaptive immune response work if a person is reinfected with COVID? If it works, then what is the lifespan of antibodies generated?
Right. Now, it's again an interesting question, but the thing is that this reinfection is not proved. Let me tell you, the reason is that if the virus is infected, normally everyone knows that it will be there for 7 days. After that, the antibody will be formed. But see what you are testing, either an antigen testing or you are doing RT-PCR. Now, viral particles will be there. Now, viral particles has been shown to be, or dead virus is shown to be present up to 3 months time. Now, what you might be testing in RT-PCR or antigen testing is probably an existing one. Nobody has proved after 2 or 3 months time live virus is present or not. So I doubt about that reinfection that is to be proved probably, unless the virus has got mutations takes place. You are coming out with a new protein within the same virus, then it's a different thing.
Yeah, but there is also one possibility which has been shown in other virus. Even if the reinfection happens, it will be mild. It will not be very severe and tough.
Correct.
Yeah.
Okay. Do we have time to ask one more question, Sanjit?
No,
quickly.
Sabrish. Okay, take one question, Sabrish, and then.
Okay, this is the last question.
Next speaker.
Yeah. Okay. One last question. Azithromycin is known to inhibit the spike protein CD147 interaction. But clinically, hydroxychloroquine azithromycin combination was not effective. Can we assume meplazumab, that is anti-CD147 antibodies, won't be effective as well?
Right. I have mentioned about 147. That is basically that antibodies for the purpose of diabetes and asthma. I'm not sure whether it has come out or not. Probably it might be useful. Probably it might be useful. Probably. Because, if you remember in one of the slides I have mentioned about the CD147 antibody, how that is involved in the interaction with the corona's entry into the host cell.
Right. So I hope the participant name is Madhu, whose question is answered. Thank you, Dr. Ramchand, for a very informative and very nice talk.
Thank you, Dr. Ramchand. So thank you again. I think we will have certainly a lot of interaction and share our
Thank you, Professor
efforts. Yeah.
Professor Sabriz, and especially to Professor Krishnan. Thanks a lot for this opportunity. Thank you very much.
Thanks very much.
I will stop my sharing of the slide.